Meet Megan and Chris. They're in their mid-30s and have been talking about getting kids for some time now. Well, they're two very healthy individuals that both have relatives that suffer from a disease called cystic fibrosis, or CF. CF is a recessive genetic condition, meaning that two unaffected carriers have a 25% chance of getting a kid together with CF. CF patients have problems producing mucus, and this especially causes a lot of trouble in the respiratory, digestive, and reproductive organs. They need lifelong medical support and have a life expectancy of around 40. Because CF is relatively prevalent in their family, Megan and Chris decided to get their DNA tested to see if their kids would be at risk. 3 weeks later, they hear that they are indeed both carriers of the gene. So what do they do now? They would really like kids, but are worried about the 25% chance of bringing a kid into the world with CF. They know that testing is possible when Megan is pregnant for 10 to 20 weeks, but she would not like to undergo abortion anymore at this stage. So can they make sure they conceive a baby without CF? Absolutely. The answer is pre-implantation genetic diagnosis. For pre-implantation genetic diagnosis, fertilization has to occur outside the body. We call this in vitro fertilization. And what happens is that the ovaries are stimulated with hormones to produce several egg cells at once. These are then harvested and fertilized with sperm cells in the lab. Each fertilized egg results in a zygote, the one cell that will become the entire newborn. This cell contains the complete DNA sequence of the potential baby. One day after fertilization, the zygote divides into 2 cells called blastomeres. And on day 2, they each divide again, resulting in 4 cells. By day 3, there are 8 cells, and this lump is called a morula. This is when pre-implantation genetic diagnosis is performed. What happens is that one of the 8 blastomeres are removed from the early morula and used for DNA testing. By looking at the DNA in this one cell, researchers can tell whether the developing embryo has inherited cystic fibrosis or not. With the extracted cell, researchers can find out many things about the potential baby, and to do so, the DNA must be analyzed. There are 3 ways to do this. 1. Gene sequencing. The DNA can be sequenced in order to obtain all the genetic information about the embryo. This is more expensive, relatively prone to error, and is also often unnecessary to have all this information. 2. Chromosome mapping. Mapping and ordering all the chromosomes of the extracted cell can discover abnormalities in the number or the configuration of chromosomes. This will provide information about, for example, Down syndrome and the sex of the embryo. In some countries, this is included in all in vitro fertilization procedures. Lastly, which is often the preferred option, is screening the embryonic DNA for specific mutations that have been identified in the parents already, such as the cystic fibrosis genes in Megan and Chris. The extracted cell's DNA is normally identical to the DNA of the other cells in the morula, which is why these last 2 techniques have an accuracy of about 99.5%. Human error in the lab is always possible, but it's probably more likely that couples have had unprotected intercourse around the treatment period, which must have fertilized an egg cell that was not harvested in the first place. But still, accuracy is pretty high. So let's get back to Megan and Chris. Because a couple of Megan's egg cells have been harvested and fertilized, at least one of them is likely to not possess the genetic combination that leads to CF. This embryo is selected and the other ones are discarded or frozen for future use. On day 5, the 7 remaining cells have grown to around 100 cells in the lab, which is when the early embryo is placed back into Megan's uterus, where it will have a 20 to 30% chance of developing pregnancy. If unsuccessful, Megan will just get a period after a couple of days, as always. But removing 1 out of only 8 cells, isn't that detrimental for the development of the embryo? Apparently not. The newborn will be just fine. Even though the early embryo is in its most fragile state this early in development, removing half the cells could still lead to a healthy baby, because twins also develop at this point. So for a large Belgian study titled Neonatal Follow-up of 995 consecutively born children after embryo biopsy for pre-implantation genetic diagnosis has concluded that there were no negative consequences of the physical and psychological health of the newborns. However, we ought to be careful in the interpretation of studies as such, because this technique has barely been around for 1 generation. But even though the long-term consequences are still considered to be unclear, pre-implantation genetic diagnosis definitely seems like the best available option for Megan and Chris at this point.